Good afternoon, everyone. The Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of our Biochemistry Focus webinar series. So topics in this series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar gives the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in the series. Please see the website for more details. I'm Ian Collins, Professor of Medicinal Chemistry at the Institute of Cancer Research in London, where I head our chemistry department in the Multidisciplinary Cancer Drug Discovery Centre. I'm delighted to be able to chair this afternoon's session. So this event is part of the Biochemical Society's dedicated early career research programme of webinars, and we're delighted to provide this opportunity today for Louise, Nicolas, Bethan and Deepak to share their work with the biosciences community. Our webinar today is titled Developments in Drug Discovery, and we will hear from these four early career researchers who will share their work in this field. Before I hand over to the first speaker, I'd like to mention that questions will be asked at the end of the webinar when all four speakers have presented. But please do send in your questions by text during the talks. If you have a question, please type it into the question box as shown in the image on the screen, stating who your question is for, please, and we will try to answer as many of those as possible at the end. So our first invited speaker today is Dr. Louise Walport from the Francis Crick Institute, London and Imperial College. So Louise obtained her doctorate under the supervision of Professor Chris Schofield and Professor Christina Redfield at Oxford, focusing on mechanistic studies of histone demethylases. Following further postdoctoral work in Oxford, she was awarded a Marie Skłodowska-Curie Global Fellowship to work in the group of Hiroaki Suga at the University of Tokyo, where her interest in cyclic peptides arose. Since 2018, she's been a lecturer in the chemistry department at Imperial College and also a group leader at the Francis Crick Institute. Her group is particularly interested in developing new approaches to understand the regulation of enzyme-catalyzed post-translational modifications using cyclic peptide-based tools. And today, Louise is going to talk to us on diverse binding to a single interface, de novo cyclic peptides as inhibitors of the BET bromodomains. Louise, please give your presentation.
Thanks, Ian. Good afternoon, everyone, and thank you for giving me this opportunity to share with the Biochemical Society community some of our recent work. As you heard from Ian, overall my lab is interested in developing chemical tools based on cyclic peptides, particularly to understand the regulation of proteins involved in post-translational modifications. And so why are we interested in peptides? Well, I think peptides are emerging as a powerful class of drug-like molecule for targeting challenging drug targets, things like protein-protein interactions or members of highly homologous protein families, places where classical small molecules have maybe found it quite challenging. And this is partly due to their size and dense functionality. So I like to think of them as occupying this sort of Goldilocks region in space between large biologics, things like antibodies, and classical small molecule drugs. And this allows them to adopt favourable characteristics of both classes of molecules. So like biologics, they have really exquisite target specificity and potency, and are largely made up of amino acids, so break down to benign metabolites. However, in contrast to biologics, their small size means there's little risk of immunogenicity, and they can penetrate tissue and also in some cases cell permeability, that this is more challenging, and the manufacturing costs tend to be lower than for biologics. The other reason we like them is there's a lot of really good technologies for discovering peptides. And in my lab, we apply an mRNA display-based system called the RAPID system that was developed in the lab of my postdoctoral advisor, Hiroaki Suga at the University of Tokyo. And the RAPID system allows us to produce encoded libraries of peptides with over 10 to the 13 different members that we produce using in vitro translation and genetic code reprogramming. So we can make cyclic peptides with a large number of artificial amino acids in them. And we can take these encoded libraries and select from them tight binding peptides to a target of interest by using affinity panning with an immobilized protein. And we repeat these rounds of making our library and selecting peptides until the library becomes enriched in sequences that bind to our target. At that point, we can use next generation sequencing of the nucleic acids that encode these peptides to identify the sequence of our hit peptides. And so really in just a few weeks, we tend to be able to go from these trillion member libraries to just a few tight binding peptide sequences, often in the nanomolar or even picomolar range. And these libraries are really powerful. You can almost always get hits to proteins. And what in this study we were interested in understanding really is what the Greater structural diversity is in these libraries, whether you have more than one different structural class that can bind to a given target. So really what's underlying the success of these libraries. And we decided to study this in a collaboration with Joel Mackay's lab at the University of Sydney, looking at bromodomains, which are small alpha helical bundle proteins that recognize acetylated lysines. And we chose to focus on the Bet bromodomain family, these 4 proteins, BRD2, 3, 4, and T, which are transcriptional co-regulators that recognize acetylated lysine in a range of positions on histone tails and in transcription factors. And in so doing, they regulate gene transcription. And it's found that these proteins are frequently misregulated in cancers. And indeed, already there are more than a dozen clinical trials ongoing for drugs targeting the bromodomains of these proteins for things like acute myeloid leukemia and ER-positive breast cancer. But despite over a decade of medicinal chemistry efforts targeting these proteins, it's been very challenging to get small molecules that can either distinguish between the first bromodomains and second bromodomains of these proteins, or even more so between individual bromodomain ones of a given family. And when you look at this overlay, this is an overlay of crystal structures of 3 of the first bromodomains bound to one of the archetypal inhibitors here in yellow, JQ1. You might see why, and that's because around the pocket that replicates the acetylated lysine, really all the residues are conserved. So it's been difficult to pick up interactions that are specific to an individual protein. So we wondered whether cyclic peptides might be able to pick up differences that these small molecules have, and whether, because these are quite crystallographically amenable, we might be able to understand how this targeting is coming about. So I set out to carry out 3 rapid selections against the first bromodomain and the second bromodomain of BRD3, and then also the second bromodomain of BRD4. And what we were immediately happy to see is we got lots of peptide hits from here, and here you can see just 3 examples where we measured their affinities to each of the bromodomains using SPR. And immediately we could tell that we could get very potent hits. These are all in the nanomolar, or in some cases the picomolar range. And not only are we able to get potency, but we've really got great selectivity. So for example, this peptide here, 3.1b, binds in the sub-nanomolar range, the first bromodomains of these proteins, and really doesn't bind at all to the second bromodomain. And that's really much greater selectivity that's been able to be achieved with small molecules. And not only that, but we're beginning to be able to get some selectivity even within the first bromodomains, although we've got a way to go before we've got something that hits just a single bromodomain here. We also did some commercial assays where they screened these against a wider family of bromodomains. All these proteins in bold here with the yellow circles, and red show hits for inhibition, and you can see that we're selective not just within the BET family, but also the broader range of bromodomains. But then the question really arises, how are we getting this? And so we did some structural work, and this was led by a very talented PhD student in Joel's lab, Karishma. And the first crystal structure she was able to get was this one here with peptide 3.1c. And you can see we have a really nice compact structure with an acetylated lysine that points down into the binding pocket. And when we compare that to a natural ligand here, a histone 4 binding to the same bromodomain, you can see that we're really mimicking the same interactions. We have an acetylated lysine in our pocket that's able to pick up the natural hydrogen bond with this asparagine here. And that's very nice, but then it made us think, given this, why are we not getting the same interaction with the second bromodomains, given we seem to be mimicking this natural interaction? So Karishma then set out to try and crystallize these with the other proteins, and luckily she was able to. So for the first bromodomains, you can see we get an almost identical overlay in all 3 cases, and that's consistent with their similar binding affinities. But for the second bromodomain, to which we bind 2 orders of magnitude less strongly, we were initially surprised to see that maybe the binding modes looked pretty similar. We're still binding in this main pocket. But when we looked a bit more closely and more distal to the site, around the back here from this angle, what we saw that there was another cleft in the first bromodomains formed by this phenylalanine here and the aspartic acid that forms a groove in the protein surface into which we had a second acetylysine sitting, picking up interactions. And in those second bromodomains, this phenylalanine is a tyrosine instead. So there's no longer that groove, and we lose the interactions with this acetylated lysine, which then points back out into space. So we think these interactions distal to the binding pocket are what's giving us selectivity with peptides that isn't achieved if you just focus with a small molecule around this site. So then the question is that a conserved motif with all our peptides? And the answer was no. So we've got a lot of different things we saw, and the next one we saw was this very interesting bivalent binding mode. So this is a 2-to-1 mode, and we really think this is a fairly ternary interaction. So on these surfaces, you can see we've got interactions between the peptide and the bromodomains, but also between the two bromodomains. And here, how we derive selectivity was clear, because when we tried crystallizing this with a second bromodomain, we see we now get a 1-to-1 complex. And so we think in terms of drug discovery, this might be another way to get selectivity by trying to make a peptide that binds to the first bromodomain and the second bromodomain of an individual BET protein. The other interesting point we thought was that these are really highly divergent binding modes. The only similarity between our two peptides is that they both have an acetylated lysine pointing down into the acetyl lysine binding pocket. And that was a common theme. So we observed a lot of other bivalent binding modes. You can see 3 here. We also saw helical modes. And I guess it now looks like I'm just throwing structures at you, but it's really to emphasize my main point, which I'd like to make. And I think we think that maybe this has some implication for drug discovery more general when we think about peptide design. Because if you don't use de novo libraries, another way of synthesizing peptide inhibitors is to make something like a staple peptide, where you take a natural ligand and try and stabilize it. And really, there's a lot of structures of bromodomains here with natural ligands all overlaid. I don't think we'd have thought these were good candidates for making something like a helical staple peptide, because all the natural ligands bind with no secondary structure. And yet, when we look at our de novo peptides, what we find is that actually the same interface can bind a whole different set of beta hairpin peptides. Along the same interface and groove, we can also get helices. And so really, you've got a single binding surface here that's poised to bind many different secondary structure elements. And so maybe these de novo peptides for unstructured binding partners might be good starting points that you could then convert, even if you wanted to, into helical mimetics. And even if you've got a helix of a natural ligand binding, maybe sometimes you want to think about hairpins as an alternative binding mode. So with that, I'd just like to thank all of my lab members, some of whom you can see pictured here, back in pre-COVID days, all of Joel's lab, particularly Karishma, my other collaborators, the scientific technology platforms here at the Crick, all of our funders, and you for your attention. I'd be happy to discuss things at the end of this session. Thank you.
Thank you. Thank you very much, Louise, for an excellent start to our session this afternoon. Just a reminder to everyone listening that questions will be asked at the end of the session. Please send questions by text. And I'd like to thank Louise and move on and introduce our second speaker today, who is Dr. Nicolas Bery from Inserm, Université Paul Sabatier, and the CNRS and Cancer Research Centre of Toulouse. So Nicolas is a senior postdoctoral scientist at the Cancer Research Centre of Toulouse, where he implements intracellular antibody approaches to define novel therapeutic strategies against difficult to drug proteins in pancreatic cancer. And today he's presented his work titled The Discovery of Potent KRAS Macromolecule Degrader Specifically Targeting Tumours with Mutant KRAS. Nicolas, please give your presentation.
So I'm going to present the work I've done. Actually, I'm working at the Cancer Research Centre of Toulouse, but I'm going to present the work I've done in Terry Rabbit's group on targeting KRAS with macromolecules. So why we would like to inhibit RAS and KRAS? Well, RAS proteins are mutated in around 20% of cancers, and it would be valuable to target specifically KRAS, as it represents more than 85% of RAS mutations. However, so far, most of the current inhibitors target the conserved effector lobe, shown here in wheat color, of RAS, and that is 100% conserved between the different RAS isoform. Therefore, as an alternative, probably targeting the allosteric lobe, which is the other side of the protein, shown here in gray, might be a possibility to inhibit RAS function in cells and to reach the isoform specificity. So we started the project in collaboration with 2 groups from AstraZeneca, and they performed the phage display selection with a DARPin library against KRAS G12V protein. So a DARPin is an antibody mimetic protein that is able to bind antigens or targets inside the cells or also outside, and we used that scaffold to bind KRAS. And what we obtained among the binders, we obtained the DARPin K19, shown here in yellow with color, that binds KRAS on the allosteric lobe, shown here, and the interface is shown here in blue. So it interacts on the alpha-3, loop 7, alpha-4 region of KRAS, remote from the switch region. And it has an affinity of around 10 nanomolar for KRAS, and it's able to bind to KRAS GDP and KRAS GTP. Next, we wanted to check whether this DARPin could bind all RAS isoform or not. So to do that, we used our bread-based RAS biosensors we previously developed. These biosensors monitor RAS protein-protein interaction in cells. So what we observe is in cells, when the DARPin K19 was able to interact with KRAS wild type, it's the gray curve, but also with KRAS mutant, the black curve, but it's not able to interact with NRAS or HRAS wild type or a mutant, shown here in orange and blue curves. So then we wanted to, we then said that the DARPin K19 binds specifically to KRAS, and we wanted to check how it could get this specificity. So after analyzing the interaction map of the DARPin on KRAS, we observed that this DARPin was binding two KRAS-specific residues, the histidine 95 and the glutamic acid 107. So we decided to mutate these residues by the one found in NRAS and HRAS and test the ability of the DARPin to bind to these mutants in cells using the BRET assay. And again, what we observe is the DARPin K19 binds to KRAS wild type in orange, but also binds to the E107D mutant in black. However, the DARPin does not bind to the histidine 95 mutants, which means that the K19 binds specifically to KRAS via its interaction with the histidine 95 residue on KRAS. After getting a KRAS-specific residue, we thought to engineer it into a single domain-based degrader. So these degraders are basically our targeted protein degradation that we use with intracellular macromolecule binders. So basically, or an intracellular antibody or antibody mimetic, such as a DARPin, is fused to an E3 ubiquitin ligase domain. And by binding, when the intracellular binder contact its target, this target will be ubiquitinated and degraded by the proteasome. So we used, after several rounds of optimization, I engineered a pan-RAS degrader by fusing an intracellular single domain antibody that binds to all RAS isoform with an E3 ligase domain, and I've engineered the KRAS degrader by fusing the DARPin K19 I just talked about with the VHL E3 ligase. What we've done then is to transduce the cells to express these degraders inside the cells, and we did several cell lines. I'm going to show you the data for two different cell lines. One is a KRAS mutant cell line, and the other one, MRC5, is a RAS wild type cell line. We first checked whether the pan-RAS degrader was able to degrade its targets. And what we observed by Western blots is the depletion of all RAS isoforms by Western blots in both cell lines and in all the cell lines we've been tested, which are KRAS, NRAS, and HRAS. And then we check the KRAS degrader. So once we induce its expression with the doxycycline, we observe that the KRAS isoform only was degraded, but not NRAS or HRAS in all the cell lines we tested. Then we showed that these both degraders were able to induce a rapid and sustained degradation of their target in cells. So as you can see here, only after only 2 hours of addition of doxycycline, which promotes the expression of the degrader, we could see a decrease of KRAS protein by Western blot, and that decrease is sustained in the time, in this, as you can see in this time course experiments. Then we wanted to check the effect, the effect on RAS downstream signaling pathway in these cell lines when we express the two different degraders. So what we observed is that the pan-RAS degrader, when it's expressed, inhibits RAS downstream signaling pathway, as shown here with phospho-AKT, which is part of the PI3K pathway, but also it inhibit phospho-MEK, phospho-ERK, which are part of the MAPK pathway downstream of RAS. And this is, we observe this in all the cell line we've been tested, regardless of the RAS mutational status. However, what we observe with the KRAS degrader is an inhibition of RAS downstream signaling pathway only in mutant KRAS cell lines, but not in here RAS wild type or NRAS or HRAS mutant cell line. So here there is no inhibition of the RAS downstream pathway, even though we are degrading KRAS in this cell line. So the next step was to assess the functional effect of these degraders in the cell line proliferation in 2D in vitro assay. So in the mutant KRAS cell line, both KRAS degrader and pan-RAS degrader were able to inhibit the proliferation of that cell line, while in the RAS wild type cell line, only the pan-RAS degrader was able to inhibit the proliferation of that cell line. Finally, we wanted to check whether these degraders were efficient in vivo. So to do that, we made two different cell lines, which are the H358. It's the cell line I showed you that is KRAS mutant. And this cell line is expressing the Firefly luciferase to be able to observe the tumor cells in the mice. And it expresses conditionally or either the KRAS degrader or the pan-RAS degrader. And each of these cell lines have been injected in mice. 18 days later, when the tumor were formed, we started the doxycycline treatment for 20 days. And we measured the tumor growth over the time. And what we observed is day one is when we started the doxycycline treatment. So you can see that the group of mice that were not treated, the tumor was still growing, which is shown here in orange and in black, while when we started the doxycycline treatment, which induced the expression of the degraders, we observe a regression of the tumor growth over the time. So what we observe during in that work, and the conclusion is that from a phage display selection of DARPins against KRAS G12V protein, we isolated among the binders one KRAS specific DARPin that has been engineered into a KRAS degrader that is able to degrade KRAS wild type and mutant proteins, but to only affect and inhibit in vitro and in vivo mutant KRAS cell lines, mutant KRAS cells and tumors. So here we show the proof of concept for the development of KRAS degraders as therapeutics that could be implemented to any tumors that would have a KRAS mutation. So I would like to thank the people from Terry Rabbit's group and the two groups of the people from the two groups of AstraZeneca that were involved in that work.
Thank you very much, Nicolas, for an excellent, clear presentation. As I remind people that we'll have questions afterwards, so I'll thank Nicolas and move on to our third speaker today. So that is Bethan Howells from AstraZeneca. And Bethan joined AstraZeneca in 2016 after completing her undergraduate biochemistry degree at Leeds University. She started her career as a microbiological lab technician working in the quality control operations, but in 2017 moved into research and development to work as a research scientist in the high-throughput screening group. And in 2020, after 3 years in that team, Bethan moved again into the protein science team as a DNA-encoded screening scientist. Her main role over the past year has been in the setup and implementation of DNA-encoded library screening at AstraZeneca, part of the technology transfer of the DEL screening from XChem Therapeutics. So Bethan now works as a senior research scientist in the protein scientist team, completing DNA-encoded library screening for drug discovery projects and also identifying novel applications for this exciting technology. And today, Bethan will present her work in this area. Bethan, please give your presentation.
Thank you, Ian, for my introduction, and thank you to Biochemical Society for the opportunity to present to you all today. First of all, I'm going to explain briefly what DNA-encoded library screening is, and then I'm going to go on to talk about how we're using it currently in AstraZeneca. So firstly, what is DNA-encoded library screening? So DNA-encoded library screening is a hit identification approach, and the aim of a hit identification approach is to find hits. So hits are compounds which will bind and modulate the activity of a target of interest. And these are either recombinant proteins or cells in vitro. And then these hits can be inhibitors or activators against a therapeutic target. DEL screening focuses on finding hits from a compound library. And we want to find hits which bind to recombinant protein targets. We can then assay these hits to ensure they have the desired mechanism of action. And these hits can then be developed into lead molecules within a drug discovery campaign and ultimately into drugs which will benefit patients. So the first step is to synthesize the libraries. So in library synthesis, building blocks are added sequentially to form a compound. As each building block is added, a DNA tag which encodes the associated building block chemistry is also added. As more building blocks are added, so are additional DNA tags, and then the complete compound will have a DNA tag attached to it, which acts like a barcode to denote the associated chemistry. This allows us to then pool all these compounds together to form a library, and a library can contain billions of compounds. We then take these libraries and incubate them with the recombinant protein target of interest, which will contain an affinity tag. Any compounds from the library which bind to the target protein will be immobilized when we immobilize this target protein and co-captured with it. Any compounds which don't bind are washed off in stringent wash steps. And then we can take what's left and elute it from the target protein. So we elute these bound library members, cleave off the DNA tags, undergo PCR amplification to get to a suitable DNA concentration, and then we send this output for high throughput sequencing. The sequencing information can then be decoded to give all the information on the enriched compounds. And it's worth noting at this point that compounds which we find to be enriched must then be resynthesized without the DNA tag for any follow-up testing. I'd like to compare this to another hit identification approach, specifically high throughput screening. So DEL is just one of many hit ID approaches, and we actually usually run DNA encoded library screening in parallel with others such as high throughput screening, fragment-based lead generation, and virtual screening. But to compare it specifically to high throughput screening, in high throughput screening, we use quite a lot of automation to screen millions of compounds in an arrayed screening approach. So this means that each of the individual compounds in a high throughput screen is in an individual well of a microtiter plate. So in DNA encoded library screening, we don't use an arrayed screening approach, we use a pooled screening approach. And this means that within one PCR tube, we can take billions of library members and our target protein and do the screen in just one tube. For this approach, we only need 2 mg of protein. And to compare this to high throughput screening, a typical high throughput screen can use tens to hundreds of mg of protein, depending on the project and the assay developed. In DNA encoded library screening, we also have the advantage of being able to screen 12 conditions in parallel. And these conditions are usually all linked to the target protein. So the first condition you may have will just be the target protein in isolation. You can then include mutated forms of your target protein or truncated forms. You can also include your target protein with any relevant binding partners. And you can also include some off-target conditions too, to get an early indication of the selectivity of your hits. In high throughput screening, you generally just screen 1 condition per campaign. One of the caveats of DNA encoded library screening is that it is just a binding assay, so we only find binders. Within a drug discovery project, you want to find compounds which will bind and then alter the activity, so additional assays are required downstream of DEL screening to confirm the activity that we have here. Whereas comparatively to high throughput screening, you usually run as a primary assay an activity assay, so you already know a bit about the activity of the compounds you find. I want to briefly touch on AstraZeneca's history with DNA encoded library screening. So DNA-encoded library screening is a relatively new approach compared to some other hit finding methods. And at AstraZeneca, we have quite a long-standing partnership with a company called XChem Therapeutics over in Boston, and we outsourced our DNA-encoded library screening to them for many years. And this has been a really fruitful partnership. We've been able to license many compounds through this, and we've had an impact on numerous internal drug discovery projects, one of which is highlighted here. In 2018, we actually underwent a technology transfer, and this means that XChem agreed to train us and set up the platform in-house at AstraZeneca, and our partnership with them would come to an end. So 2 years ago, in 2019, we began training AstraZeneca to bring this technology in-house, and last year, in 2020, was the first year that we had a full year of DEL screening at AstraZeneca. So last year we had three main objectives or applications for DEL screening. The first was a relatively simple objective, and this was just to support projects. And by this, I mean the drug discovery projects within AZ. So we were to take our training and the methods we learned from XChem, and we wanted to use this to support projects and deliver equity to live projects, which we were successful in doing. We then had two more interesting objectives, and I'll touch on these in more detail. So the first of these is multi-target screening, and then the second is process development. So multi-target screening, as I mentioned before, we have up to 12 conditions within a DEL screening campaign that we can use. And these are usually used to screen different conditions that are all associated with the same target. But we thought, what if we use these 12 conditions to screen 12 bespoke targets in one screen? And we thought of two main applications for this. The first one was to build our internal PROTACs toolbox. So a PROTAC, if we look at the diagram here, is a two-headed molecule which is joined by a linker. One of these heads is a ligand which binds to your protein of interest, and the second head is a warhead which will selectively bind to an E3 ligase. The E3 ligase, which then becomes bound to your PROTACs, can then tag your target protein for ubiquitination, and this will cause it to be degraded by the proteasome. So PROTACs are a really useful tool to degrade unwanted or harmful proteins with very high specificity. So in AstraZeneca, we decided to use DEL screening. We chose multiple E3 ligases which were of particular interest to our therapy areas and screened these in parallel. So this meant it was really efficient because they were all screened at the same time, but it also gave us the benefit of being able to use each E3 ligase as an off-target for all of the others, which gave us an early indication of the selectivity of the hits. Any hits that were identified can then be resynthesized without the DNA tag, and this will then form the E3 ligase warhead, and this can be fed into our PROTACs toolbox. This PROTACs toolbox allows us to have the materials ready to build specific PROTACs very quickly, and hopefully generate degradation data like the example seen here. A second application we thought of for multi-target screening was to provide feasibility assessments prior to starting projects. So usually within AstraZeneca, before a drug discovery project is initiated, a lot of validation work goes into assessing the feasibility of the project before we put resource into a full drug discovery campaign. So for some riskier targets that have limited validation work and no tool compounds available, we thought DEL could be an opportunity to assess the feasibility. So firstly, we identified some targets based on a defined criteria. And then we took these targets and we went into protein production. So we completed expression tests for the recombinant proteins associated with the targets, and then we purified any suitable constructs. We then completed a DNA-encoded library screen of all of these targets in parallel. And from the data analysis, we're able to get some indication of the druggability of these targets. So can we find any binders to these? And then also, if we identify any hits and confirm these without the DNA tag, we can then feed these back into the project for use as tool compounds. The third objective was around process development. And the process development focuses on some current hot topics in DNA-encoded library screening. The first one I want to mention is membrane protein screening. So recombinant membrane proteins are notoriously difficult to express and purify in high quantities. And as DEL screening only requires a small amount of protein, it's seen as quite a good option for getting around the difficulties with screening membrane proteins. So there's this paper that's been published in Nature Chemistry last year, and this describes a method for screening membrane proteins on the surface of live cells. And this explains some really interesting approaches, but it's also got some very specific requirements and bespoke methods. So this adds other challenges if we try and adapt this process, but this is definitely something we're looking at bringing in-house. Another area for process development is around DNA binding proteins. So DNA binding proteins are becoming more increasingly common as targets, but they come with quite a lot of risks in this method. So to try and explain this, within a usual DEL screening campaign, you have your library members, so you have your compound with the DNA tag associated with it. And what should happen is you have your target protein, and the compound should bind to your target protein. The problem with DNA binding proteins is that they have DNA binding domains, and the risk is that the DNA tag from your compound might bind non-specifically to your target protein. And this will just look like a hit. So this will be a false positive. And it's really difficult in the data analysis step to differentiate between a true hit and a false positive. One of the ways you can get around this is purifying your protein without the DNA binding domain, but this isn't physiologically relevant, and actually not all proteins are stable without them. So what we're currently looking at as well, and we have an industrial placement student with us this year, is methods to screen DNA binding proteins without removing the DNA binding domain. And then just to touch on some future visions, so one of the main things we're focusing on is really improving the efficiency of this process. The current timelines to get compounds back to project teams is about 9 months, but we think we could make this faster. Implementation of artificial intelligence and machine learning is one of the ways we're looking into doing this to really decrease some of the timelines associated with screening. And we're also always looking for continual method development. I've mentioned a couple of things we're looking into, but we want to know how we can screen more challenging targets in more challenging conditions. Thank you very much for listening to me today, and I also want to acknowledge everyone who's involved in the DNA-Encoded library screening process. We have a team of biologists, sequencers, chemists, and then QBio, and people helping us with machine learning and artificial intelligence.
Thank you very much, Bethan, for a really clear and really insightful talk. Just to remind everybody, the chat function is open for questions. There's a few come in already. Please continue to send those in. And I'll thank Bethan and now move on to our final speaker of the day. So this is Dr. Deepak Balaji Govindaraj from the Council for Scientific and Industrial Research Biosciences in South Africa, where he's Center Manager of the CSIR Synthetic Biology and Precision Medicine Center. So Deepak is an expert in nanobiotechnology, synthetic biology and cancer precision medicine. And he's currently establishing cancer precision medicine as a platform at CSIR in South Africa. And delighted to ask him to give his presentation today on ovarian cancer for precision medicine. Deepak, over to you.
Thank you very much. Thank you very much for the opportunity for the presentation. I'm going to present about precision medicine, which is on the approach of using drug repurposing for cancer precision medicine, which we are currently establishing at Council for Scientific and Industrial Research in South Africa with the support of our Department of Science and Innovation. And we are going to particularly focus on the cancer called as ovarian cancer, and we use synthetic biology approach to enable for drug repurposing in precision medicine. What is precision medicine? It is far more important to know what person the disease has than what the disease the person has. So it's very important to know what person the disease has, and that's where the personalized precision medicine approach works. And why we are particularly focusing on cancer is that cancer is one a deadly disease, and it's growing in numbers, high numbers in South Africa. And it is expected by 2030 there will be 78% increase in cancer in South Africa. And ovarian cancer is one of the top 10 cancers for women. And when you compare the cancer in number from between South Africa and across the world, it is irrespective of the Human Development Index, the ovarian cancer has a deadly impact across all the populations in all the continents. By addressing the unmet medical need for ovarian cancer in South Africa, we are also able to address the unmet medical need around the world. And also to give the relevance why we are working on ovarian cancer is that ovarian cancer in 2011 was close to 7% of among the female cancer deaths. By now, it has reached as closer to breast cancer and lung cancer with close to 24% in percentage of cancer deaths in women. And in addition to that, there is no cure for ovarian cancer, although we know that only 5 to 10% of all cancers are inherited. However, every woman is at a risk of developing a gynecological cancer. So it is very important to solve this unmet medical need. There are so many ways to solve this unmet medical need. And cancer having be a guardian knot with complex unsolvable problem, we can either do a classical drug discovery approach and solve that problem, or we can use an unconventional approach like Alexander the Great did by breaking the guardian knot by just cutting the knot by a knife. Such kind of an approach is something we are currently implementing, is to use a precision medicine approach by using drug repurposing. So how this approach works is that basically we have a panel of drugs, of already approved drugs for all types of cancer, and we use patient samples from ovarian patient samples and test those drugs on those patient samples in a 96 or 384 well format and see whether those drugs are effective in killing those cancer cells in vitro setting. And we will further validate those drug candidates through flow cytometry. And then, because it's an already approved drug, we bypass the strategy of doing clinical trial or xenograft experiments and thereby cutting the time frame required in order to enable patient benefits. And we have 3 work packages. The first work package is where we do the high throughput drug screening, where we culture the cancer cells in a 384 or 96 well format with already approved drugs, and then identify drugs which are effective in killing those cancer cells in a short time frame. The second approach is to use microfluidics, where we capture cancer cells in a well with few drugs to identify single drugs which could be effective in killing drug-resistant cancer clones. And the third work package is to study how the tumor microenvironment impacts the drug effects on the cancer cell. And because it's an already approved drug, the potential drug candidates which I identify, we directly go for precision cancer therapy. One of the major advantages of using our drug repurposing platform is that the conventional approach where we do drug discovery, it's a long and lengthy process which can take to 10 to 17 years. By using drug repurposing platform, where we cut down the time frame by close to 5 to 6 years, we enable the patient benefits where the drugs we identify using our treatment platform could directly benefit the patients, and also we could expand this to several hospitals and use it for clinical testing. And we have a 5-year time frame to bring this platform to a market strategy for the whole South Africa in a 5-year time frame. Besides these work, we have an additional platform which is called the synthetic nanobio machines platform, which add on support to the cancer precision medicine, where we produce biopharmaceutical drugs using CHO cell expression system and synthetic viral platforms, and those drug candidates are tested in our cancer precision medicine platform. In addition, we improve the drug efficacy by doing nanoformulations using nanotechnology approach and thereby integrating synthetic biology to cancer precision medicine. With this, I would conclude my presentation and thank the opportunity for the audience and for the panel members for giving me the opportunity to present the work. Thank you.
Thank you very much, Deepak, for describing your work and a really important objective there. So thank you to all 4 speakers for your presentations this afternoon. I think the organisers are going to share webcams, so we'll all be on screen at the same time. And we can now welcome questions for any of the speakers. I have a number already to look through, but please, if any occur to you during the conversation, please add those and we'll try and fit those in. If I might start, let's see, we're not quite all here yet. I was going to waiting for Louise to come back online. Hello, Louise. Louise, I had one for you to start with, which was I noticed that your BRD3 BD1 peptide had an N-acetyl lysine. So perhaps you could just explain to us a little bit more how you control post-translational modifications on the amino acids in the cyclic peptide library.
Yes, of course. So the Rapid platform we use, sorry, my screen just changed, allows us to reprogram different tRNAs using an artificial ribozyme known as flexizyme. So actually in the selection itself, I'd swapped methionine. So the methionine codon instead has an acetylated lysine. And we actually designed a library so all of our peptides would have at least one acetylated lysine there to kind of target things towards that pocket.
That's cool. Thank you. Thank you for clarifying. That's great. While we're on the subject, you also showed some interesting bivalent binders from your screen, Louise. What are you able to give some insight into the where and when that's biologically relevant, particularly in the context of these multi-BRD domain proteins? Or is that likely to be a little bit something you can only see when you've got the separated domains present?
So we're actually looking into that at the moment, and I'm not certain of the answer. I think it will be, I think in a medchem sense, it would be better if we could adapt these peptides so that they dimerized the first bromodomain and the second bromodomain of a tandem pair. But we're starting to try some cross-linking experiments to see whether, I mean, I think it's not impossible that they could dimerize. There will, along chromatin, I think, often be multiple copies of BRD3, say. So I think it's not impossible that we would dimerize it in a cellular context. It's certainly, we see it in solution. I showed crystal structures, but we've tested, and this is definitely a solution structure as well. So I don't know the answer, and we're testing that, but I think it could still be relevant.
It'd be interesting to see. Actually, it's a nice way of probing the relevance of it. Thank you very much. Some questions for Nicolas, if we may. One of our questions we've asked is just could you quickly clarify the role of the doxycycline in your experiments, because the internet became a little bit unstable while you were describing the inducible systems, and I think one of our audience would like just to clarify what was going on there.
Yeah, well, yeah, because they are macromolecules, they don't go inside the cells, unlike small molecules or even the cyclic peptides. They don't go inside the cell, so we have to transduce or transfect these as a plasmid, so the coding the DNA. So we made an inducible system. So we use a Bethan system with on the lentivirus. So we infected cells, and when we add the doxycycline, it will induce the expression of our degrader.
Thank you. Actually, related to that, looking forward for DARPin-based or other intracellular antibody technologies, what do you think are the prospects for being able to deliver those into cells, having applied them, for example, as an intravenous solution?
Well, there are several strategies, let's say, at least two main platforms, the non-viral and the viral platforms. In the non-viral, with actually the vaccine for COVID, I would say the mRNA, and it has been already used in the field of intracellular antibodies and so on. The mRNA with nanoparticles is one attractive possibility. And on the other hand, you have also the virus-based. So it's also feasible, like oncolytic viruses that would be specific of tumors, of cancer cells. That would be one way of deliver this kind of therapeutics. But it's ongoing. There is, I think, a lot to do.
Okay, thank you very much. So another question which comes in a couple of parts, but the first part is where you selected your DARPin against KRAS binding to GTP and GDP bound forms, and you got the same or similar affinity for both, I think, in the initial experiments. And then when you conjugated and linked to the component of the Cullen E3 to get the degrader, did you see if there was any influence of that fusion in the affinity for the different forms of RAS?
Well, no, we didn't check the affinity. We just checked whether they would still be able to degrade. So it only degraded the KRAS isoform. Yeah, we were not able to check whether it would degrading GDP or GTP. We presume most likely it will degrade all GTP and GTP form of KRAS only. But no, we didn't check the affinity of the engineered DARPin.
Okay, thank you. And finally, when you perform your selection in the initial library, would there be any benefit in creating a secondary library by error-prone PCR to see if that leads to a mutated form with an improved binding? Could you comment on the coverage of the primary library in terms of finding the optimum binders?
Yeah, well, yeah, it's something like usually done it's called affinity maturation, and that's usually done. We were, let's say, quite lucky because it was already quite good affinity of 10 nanomolar. But obviously, if you have a binder that is really interesting to where it's binding on the protein, obviously, but the binding is low, maybe we will have to do this error-prone PCR and affinity maturation step.
Okay, thank you very much. I've got some questions for Bethan, if I may. So first of all, is there any evidence that with on the DL libraries that you might miss some hits just due to steric crowding of the DL tag to the small molecule? And can you comment on the construction of the way that the molecules are actually loaded onto the DNA sequence?
Yes, so no, the libraries are designed with, they're going to have a DNA tag on them, so that's always in mind when they're designed. It's a little bit out of my scope, I'm not the chemist who designs them, but that's always taken into consideration. There is perhaps a limit. We try and limit how many libraries we pull together, so the libraries are generated individually with billions of compounds in, and then we can pull multiple libraries together, and there is some sort of published numbers around what is the limitation around how many compounds you should really pull together, so that you don't pull hundreds of billions, because then you may have that effect.
Okay, thank you. A question that maybe plays to other experience you've had with other high throughput screening techniques as well. What are the sort of typical hit rates for DEL against a range of targets? And a second question from that, what is the typical translation from the enrichment primary hits through to the confirmed off-DEL synthesized molecule? Can you comment on that?
Yeah, so we don't really define a hit rate in DNA-encoded library screening because it's a little bit more difficult than in high throughput screening. So in high throughput screening, a hit rate of about 1% or less than 1% is what you would go for. But again, it depends on the size of your library. So if you have 2 million compounds in a library, that's already quite a lot of compounds to then retest. In relation to DNA-encoded library screening, we generally resynthesize under 100 compounds. So considering we're screening billions of compounds, the actual rate of how many come through as binders is quite small. And then from that, the confirmation rate is usually less than 50%. So we really get down to quite small numbers from DNA-encoded library screening, despite screening billions to start with. But there's quite a diverse set of chemistry in our libraries. The diversity in the DNA-encoded libraries is more than that within our high throughput screening libraries. So we usually get hits for things, and most things do generate some hits, but yeah, the hit rate is a bit more difficult to define in DNA-encoded library screening. And
sort of related to that, what the typical size of the, if you like, the non-DNA part of the library? I know that one can do fragment screening using DELs, but in your experience, what sort of is the most efficient molecular weight range for the loaded samples?
Again, it's very hard to put an exact number on that because it completely depends on the target that we're screening. So sometimes we screen complexes, sometimes we screen just individual target proteins, and then I guess the size of the target protein, that can vary from tens to hundreds of kilodaltons. So I can't really put a number on that. They're slightly bigger than your standard small molecule because they have multiple building blocks in them. And also they vary. So some of our libraries only have 2 building blocks to form a whole compound, whereas some have 3 or 4. So again, it varies.
Okay, thank you very much. And Deepak, thank you for waiting your turn. I might ask you a couple of questions. are really taken by your use of patient samples for your screening platform. Could you sort of tell us a little bit more about where those samples come from and how you have to process them to get sort of, if you like, cell lines that can be used in this high throughput technique?
To answer the first question, we get patient samples from Steve Biko Hospital from Pretoria. So this platform is basically built. I just also have to give a brief history before that. So before we started with ovarian cancer samples, we started working with blood cancer samples, where it is more easy to access those samples because it's blood. You get a lot of cancer cells, which is fluid, so it's not difficult to isolate. You can just purify them. And we have established that platform with blood cancer samples and done the drug screening already. This was done in my previous employer's place in Norway. We have done that. So once we have shown that it is possible to do on liquid biopsies of the blood cancers, now we want to take a step further to do it on ovarian cancer samples. So we try to get ovarian cancer tumors invasively, and then we try to break down the primary cancer cells and then try to do drug screening for a period of 24 hours to 48 hours. It's much more harder than doing blood cancer samples. So that optimization is still going on. So once it's fully optimized, then we can reach the stage where we can do a proper drug screening that we have done before on blood cancer samples. And the second thing was the question about, can we do cell line drug screening? Is that the question?
Yeah.
Yes, we can do cancer cells for drug screening. However, the limitation is that this is a translational platform. So this platform actually has to benefit the patients. So whatever we do with cancer cell lines will just show the platform works in doing drug screening, but it doesn't actually benefit patient setting. So the eventual benefit comes only when we do the patient samples. So we can just do drug screening on ovarian cancer cell lines, and maybe we can identify drugs through the cell lines where those drugs are killing those cancer cell lines, and maybe we can narrow down those drugs and test it in patient samples. So it is possible to do on cancer cell lines, but our main focus is to actually benefit the patient setting.
Great, thank you very much. Well, we're coming to the end of our hour allotted for the webinar, and I'd like to thank Deepak, Bethan, Nicolas and Louise for staying on to answer your questions, and thank you for submitting the questions. Thanks to everyone for attending. And you can continue this conversation online. Please follow the @BiochemSoc or @PPP Publishing on Twitter. And if you'd like to know more about this topic, we'll invite you to read some of the related content from a variety of issues and papers published by Portland Press, which have been shared with you by the chat function during the webinar. So you may be interested in attending a related online workshop that the Biochemical Society is organising on the 12th and 13th of October this year, entitled Key Aspects of Modern Drug Discovery 2021. So it's designed and delivered by experts from industry and academia, and the training event will provide a comprehensive overview of the preclinical drug discovery process. So that will focus on small molecule target identification and validation, hit to lead and lead optimization of compounds, as well as preclinical drug development and toxicology. The online program includes lectures, case studies and small group discussions, and provides also an opportunity for delegates to engage directly with speakers. There's early bird registration by the 12th of September this year, and you can register on the Biochem website. So full details are shown on the screen at the moment. Now, we also welcome suggestions for future topics and speakers to feature in this Biochemistry Focus webinar series. So if you do have an idea for a webinar in 2022, we'd be really pleased if you could submit a proposal, and you can find more information about the webinars, propose one yourself, and also watch recordings of previous events on the biochemistry.org website. So I'd like to invite you to join in the next webinar in the series, which will be entitled Visualizing Translation of Bacteria and Viruses at Single Nucleotide Resolution, and that'll take place on Thursday, the 4th of August at 2 o'clock British Summer Time. This session will be chaired by Dr. Betty Chung from the University of Cambridge, and it aims to promote interest in ribosome profiling. And during that webinar, we'll hear from Dr. Alan Buskirk and Dr. Noam Stern-Ginnesser, who are prominent world leaders in the study of bacterial and viral translation dynamics, and have made extensive use of ribosome profiling in their work. So finally, I'd like to highlight that in these new and sometimes somewhat challenging times, it's more important than ever that we stay connected and engage with our community in biosciences. And joining the Biochemical Society's community of researchers and specialists is a great way to achieve that and to meet with fellow molecular bioscientists in the relatively safe space online. And of course, members can take advantage of a variety of benefits, including discounted registration fees for society courses and meetings, exclusive access to a range of grants and bursaries, and personal online access to journals and more. So please visit the Biochemical Society's website to find out more about that. And finally, thank you all very much to our attendees and our speakers today. It's been a pleasure to host this event. Take care, everyone. Bye-bye.